Okay, cool. Okay, let's talk about prions then. So, kind of an interesting thing, can't really call it an organism. Prions are literally infectious proteins. So, these are proteins which, by their inherent shape, can cause effects in the host animals. There's no nucleic acids, no building blocks, nothing like that, no cell wall or membranes associated with these things. But they are still able, interestingly enough, to be consumed and then affect the organism without being degraded. So they're really resistant to being broken down, which that creates a lot of issues in the, I want to make sure I have my settings off of here, sorry. Creates a lot of issues with treating these types of infections because you can't really get rid of the proteins. So kind of a weird thing. So they're proteins that when folded, these are normally occurring proteins in the cells in the organism. When folded, they do just fine. But what we see is that if they misfold, they can actually create kind of a cascade of effects, which will have devastating effects on the organism that they're in. Originally, they thought they might have been viruses or something like that, but they could never find any DNA or RNA associated with it. And what they really came down to, these guys back in the 80s and 90s figured out they were just proteins that were changing in the organisms. Now, the normal ones, the way that they label these, you have prion protein common. So if you see some of this terminology, there's a wide variety of these things, but there's a common form, and then there's what they call the scrapie form. Scrapie is one of the first diseases that they found associated with these things. But these forms, when they change from normal to the scrapie, they will start kind of a cascade or kind of a what they call in chemistry a nucleating or nucleation event. So if you ever do precipitation or something like that, these things will precipitate or nucleate out of solution. That's what happens with these proteins. It's very slow at first. So for the most part, we'll talk about this a little later. For the most part, the misfolding, you can't really see it or really see any effects from it. So we think that it takes a long time for these things to start to misbehave, if you will. But once they do, once they reach some sort of threshold, the seeding part of it, then they see kind of a rapid misfolding of more and more of these proteins and they start to build on each other. So it's kind of a, that's why I kind of think of it like a nucleation event or a seeding event, where these things will trigger more changes in proteins around them. It's really, really weird. So it starts kind of slow and then changes. The proteins themselves don't really change. So the amino acid structure doesn't really change. But what does change is the 3D structure of these proteins. And once they do, once they start to change, and it says here, it kind of goes from, if you remember your protein structure, kind of goes from a helical, helix type structure to a more beta sheet structure. And if you're curious, you can always go back and look at the structure of these things. But the beta sheets are the ones that will stick really well to each other. So they'll pancake really well. The hydrophobic surfaces will stick to each other. Once they do that, then they have a high resistance to breaking down either through heat or proteases or solvents, anything like that. You can't really get rid of the misfolded proteins. They're really resistant to removal. And if you think about these things, a lot of them occur in neurons. So getting into the brain, for example, to try to remove these things is probably almost impossible. So you can't do it with drugs, chemicals, or things like that. Once it starts, these aggregates, then they'll start to have an effect on the cells around them. So the research that I looked at is these kind of changes occur both inside and outside of the cell, but the effect is that they start to kill off the cells around them. So especially if they form outside the cells, these aggregates will start to negatively affect either the signaling or the membranes of these proteins. But the overall effect is that they start to kill them off. So here, some of the early people, when they looked at normal human brains versus these prion infected brains, you would actually see really large areas of clearing called plaques, amyloid plaques. Amyloids is a fancy word for what the aggregates. So the protein aggregates, they would call them amyloids. And that's kind of true for any type of protein that will make aggregates. They get termed these amyloid plaques, basically. Good thing, bad thing to see in your brain tissue? Huge clear, yeah, thumbs down. Huge clearing of zones in your brain. Yeah. Very, not a very good thing. You're killing off neurons. And almost always these kind of things are fatal. They call them spongiform and let's see if I can say it, encephalopathies. There you go. Encephalopathy. Spongiform because it kind of looks like a sponge. You guys know what sponges look like. So a lot of empty spots within the brain tissue. There's several different types of these things. So the first ones that we're seeing were these animal encephalopathies that could be passed from organism to organism. So I have some examples on the next slides, like scrapie or chronic wasting disease, which, by the way, we've seen in Utah and the Rocky Mountain. So this is not a third world kind of disease that we need to think about. It is in populations in the US. And so it is something to think about. Anyways, they can be transmissible in humans. Some of the ones that we've seen are Kuru, Creutzfeldt-Jakob disease, and there's a couple of other ones here. Anybody ever give plasma at Grifols? They always, you don't have to raise your hand, sorry. I can raise my hand. I didn't want to put anybody on the spot. That's one of the questions they'll ask you is if you've ever been exposed to this, or if you've ever spent time in the UK in the 80s. Most of you probably say, I wasn't even born in the 80s, right? So we can talk about that a little bit later, why they ask those questions. Of those, some of them are infectious. So Kuru, mad cow disease, these things are spread by consuming infected material. About that for a second. So weird proteins that you eat can somehow get into your brain and start to change the protein structures in your brain. Weird. So that's the large part of them the infections that they see worldwide come through the infectious route. But there are examples of sporadic infections. So these sometimes are related to either genetic changes or other random changes that you could possibly get during your lifetime. There are also some other ones that can be inherited. So sometimes you can inherit a gene with mutations that will then make you susceptible to these proteins misfolding. So almost always it has something to do with the 3D structure of that protein is being compromised. And so these proteins will readily start to misfold and then can start these nucleation events. So most of them fall into the sporadic category, about 85%. And most of these sporadic events that we see occur later in life if we're talking about humans. So it seems like just through random changes or maybe random mutations, then a lot of these things can start showing up at later. The genetic or other variations kind of come earlier in their onset, if you will. Okay, what do we start to see? Well, a lot of them are examples of neurodegenerative kind of changes. So you might get twitches or drunken walking or weakness or tremors, these kind of things. A lot of these, though, I kind of categorize them as any kind of neurodegenerative disease. There's a wide variety of things that we've seen, like Parkinson's or Alzheimer's, or let's see, what is the other generic term? Just like sorry? Yeah, thank you. So any kind of a dementia. When I think of like neurodegenerative disease, and I think this is kind of an interesting link. So you can think about Alzheimer's or Huntington's is another one. Some of them, like dementia, there's not always a clear diagnosis or or at least a clear knowledge of exactly what's happening or what's going on. With some of the other ones, like Huntington's, they have linked it to a particular protein that has mutated, but the common denominator is those proteins typically misfold. So you see a common association with prions. So with prions, those kind of diseases cause proteins to misfold. With some of these other diseases, they have linked it back to misfolding proteins. So to me, it seems like, and not just to me, I'm sure lots of people have seen the link, but it's at least some food for thought for you guys if you end up working with people like in a dementia ward or something like that. There's some food for thought here to think about what's going on. Is there a common underlying mechanism for all these types of neurodegenerative diseases? You can test for some of these things. So you can look at brain waves to see how they're impacted. You can look at other 14-3-3 is a signaling protein that is commonly used to look for disease states or other kind of changes in the brain. But I mean, I don't know if you have any experience with this, but my experience is it's not very easy to just go in and get a clear diagnosis right away. A lot of these symptoms are vague, or they're not very specific, or they show up differently in different people. So I think it's still kind of a difficult area of research. Okay, what are some interesting diseases that we've seen? Kuru is an interesting one that they found in, I think it was in Africa somewhere. Oh, it was New Guinea. There, New Guinea. But this was an interesting tribe that these people were studying, and one of their rituals was to eat the brains of their respected elders when those, especially men, I think it was, but they would eat the brains of these respected elders. And the scientists saw kind of the transmission of a particular kind of disease, which they didn't really know what it was. But it was a wasting disease that they would see that would affect the people who ate the males that died off. So they would get these headaches, joint pain, and would die within a relatively short period of time. And so this was some evidence of some sort of a disease. I don't know if these guys actually pinpointed exactly what it was, but later on it was linked to other models like scrapie. So scrapie in sheep had a lot of similarities to kuru that they saw could be passed from organism to organism. They weren't really sure exactly how. The sheep weren't eating brains, I don't think, of other sheep. But it was very specific. It wouldn't be passed to other organisms. So they tried to pass some of these proteins off to other organisms, but couldn't get it to work. So it really seems like it was species limited. Whatever form of protein that was affected, that was causing these diseases, would be limited to whatever that same type of organism was. So you could get it to run in humans, you could get it running in sheep, but you can't really pass them across. And again, they saw that these things, when they purified them, were very resistant to breaking down. Another interesting part of this is, even though we're dealing with the same types of prion proteins, it can have different effects on different parts of the brain. So to me, I don't really understand this. To me, I would think that it would affect pretty much all the proteins in the brain, affect all different parts. But depending on the disease itself, it actually affects just one, usually one part of the brain. And then that's where you see the effects from. So Creutzfeldt-Jakob affects the cerebral cortex, this familial infection affects the thalamus, Kuru affects the cerebellum. You guys get the point. But interestingly, they target a particular part of the brain, and then you see the effects from that. Okay, so this is kind of review. This shows the structures of the protein. So again, these proteins deal a lot with signaling or adhesion, so keeping the cells together. When they undergo the change, then you start having large accumulations of these proteins, and that can cause death. So here's some pictures. You can kind of see the regular protein and then these big rods that are produced from the misfolded proteins. Here's another picture as well. I stole these off the internet. Okay, looking for cool pictures stuff. But you can see how they kind of stick together and then they form these long rods. And these things are going to damage the neural tissues. Okay. Okay, let's see. A lot of this is I already kind of mentioned the phylogenetic relationship. I think this is part of where the worry is right now. You know, you might have some issues convincing people not to eat byproducts of meat or human brain or something like that. But I think a lot of the research that's going on right now is what kind of effect do these things could they have on humans if we just eat them from deer or something of that nature. Uh, that was the big scare back in the 80s, the 70s and 80s with mad cow disease. This is why they ask you if you give plasma, did you spend any time in the UK while you were here? They had this really weird extraction thing that they would use for feed. They would actually extract protein from different like meat and bone meal from cattle, and then they'd feed it back to cattle. But they found this bovine spongiform encephalopathy, so BSE, and that's what they called mad cow disease. And the scare was the human consumption of this stuff. What would that do? Could we get things from other organisms, even though we saw things like scrapie don't really come out of sheep? We've seen this with other organisms, though. I mentioned, right? What if viruses start to mutate and then have a new host range? That's kind of the scare with those kind of things, or even bacteria. Same thing with proteins, prions. The problem here is we don't really understand what changes are necessary for this to affect humans, other than the closer the protein sequence structure looks to the native host, then the easier it is for it to start the nucleation event. So the closer they look, then the more likely they're able to cause diseases. They did see some cows in other countries, and we do see this pop up every once in a while. So I looked up some data. It's kind of old, like 20 years old, but it does pop up every once in a while. And the effect of all that stuff, you know, when people stop importing meat, those kind of things, the effect obviously is going to be pretty great with the amount of money that you're going to lose not being able to sell those off. And then later on, there were some signs of what's called Creutzfeldt-Jakob disease. So this is kind of the human form of, not of Kuru, but of other prion diseases, of bovine spongiform encephalopathy. So the problem here is we think that if these things change through time, over time in other organisms, then they might be able to affect humans if those changes make it basically able to cause disease. Does that kind of make sense? So we don't really care. Well, we do care. We do care that it is happening in other organisms like Creutzfeldt, or sorry, chronic wasting disease. But we are more interested in, okay, what's going to happen if these things change in a way that then makes transmission to humans easier. So that's where we are thinking about what's going on. Okay. I would relate this the same thing with viruses, right? Viruses might only be able to affect one type of organism, but through change, then they might be able to affect humans, and that's where we want to think about. The other idea that I brought up here, though, is once they found the mechanism of these proteins, so these proteins, prions, can misfold and cause disease. What is the link then to just neurodegeneration in total? So you can add to this list all the different types of dementia or anything that is neurodegenerative. And I don't know if all of them will be linked to misfolding proteins, but at least some of them are. So I already mentioned that Huntington's, I know for sure, has to deal with misfolded proteins. So I think this is kind of an interesting link. If you guys get into health studies or research or something like that, you might run into something where they're looking at the basis for all these neurodegenerative diseases. And there's a lot of similarities. So how they aggregate, the pathology of these things, the patterns of what they target. So you see particular proteins will target particular cells and cause disease. Then the hard thing is, right, is how do we treat that? So you already know that prion proteins are really resistant to breaking them down. And you're dealing with things in the brain, which is kind of a delicate area to be messing around with anyways. Can't break them down, you can't dissolve them chemically very easily. And it's really hard to just go in and remove them like a cancer, which is hard enough. There's some interesting, I guess, interesting thoughts between that and that. Okay. So any questions on that? I'll bring up your plickers. So what are the things that we want to think about here? One is, how do prions affect host cells? And of course, we're always looking for context. Why does it matter in the scope of this class? What is the title of this class again? Micro, right? Small things that affect biology. So does it matter in the context of this class? At least knowing these things are there? I think so. That's why I brought it up. Okay, what do you guys think here? Okay, yeah, so the main damage is going to be from cell membranes, but it can also break down cell communication. So that's a good one too. And then this is interesting. What happens to most protein that you ingest in your diet? You guys who eat protein. Let's think about this. If you think back to Kuru, these people were eating food. What do we normally assume with food? Whatever it is, if it's nucleic acid, protein, fats, sugars, what happens? Did you guys take a nap? Do you learn that in anatomy? Digestion? Nutrition? Huh? Okay, Fizz. What happens? Yeah, it's supposed to all be broken down, right? But what happened with prion proteins? Obviously, they're not broken down, are they? If they were broken down into their amino acids, then they shouldn't be able to misfold and cause other proteins to misfold, should they? So this is really weird. Oh, sorry. This is really weird that these proteins can be ingested and yet somehow they make it intact through your digestive tract. Or this is true for any animal, right? Just think of a deer or a sheep or something like that. They make it intact through our digestive tract and then somehow get to the brain and they can start to cause these other proteins to misfold. So still kind of a, I guess, a kind of a black box in terms of exactly how these things are affecting the cells or getting in there, I guess. Okay, bless you. Any questions? Any questions on here, the expectations? Okay, and so this kind of goes for this rest of this week as we look through some of these diseases. I'm trying to give you ideas of these diseases and how they affect humans. And well, of course, I think it kind of goes along with the same thing we've been talking about for a while, bacterial and viruses and all those, how they affect them and how do we treat them. So here's another example of something you might run into. Okay.